we've talked about flagella, prokaryotes, and I wanted to make sure I brought up a little slide because the interesting thing is for eukaryotes, they're very different. They have different structure. We're not going to get into it. They look more like microtubules. But with prokaryotes, a lot of they're behind swimming, so it pushes them that direction as you would expect. But with eukaryotes, it's more of a pulling motion for that flagella. And so it's all so their flagella. It's also they have different places from where they start from, like the prokaryote where they add to. Prokaryotes add on one end, eukaryotes add on another end. So the growth of the flagella are different. And also they do not rotate, but they undulate rhythmically. And so it is an actual pulling motion through the media because you see it goes this way versus that way. And then cilia, I like this because it's a nice little visualization of that power stroke. So this red one right here is that power stroke. And so they're doing this whole wave motion to get that snap so that they can actually start moving things along. That's how you get that cilia moving. Okay. Pop quiz. What type of solution are the red blood cells in each of these?
Isotonic.
So can I get
Isotonic, hypertonic, hypotonic.
Hyper.
Oh, I didn't put it. So yes, isotonic, same inside and out.
Oh, that's right.
And the important part was that I said, what is the solution? So if I had said, what is the cell, then that's different. So the solution,
Hyper.
Hypo.
Well, hypo, less salt on the
Less.
outside.
Hyper.
Okay.
If you ask about the cell,
By the way. Because they
only
have
with
more
B
or
and
less.
C though.
Yeah. ISO is the same as the
They're
same.
equal.
You know, if it's the same with the cell or the solution, doesn't really matter which one you're asking about. But yeah, only for hypo and hyper.
She doesn't know.
Okay. Oh, so we've been talking about metric units of length. So I thought it was important to make sure you really looked at this and had a clue about sizes. Yeah, so scientists do everything in metric because it's units of 10 versus our system, which is confusing and arbitrary.
Moronic. It's moronic.
It's ancient.
It's from 500 years ago. We're the only industrialized nation in the world that still uses such a system.
True. But scientifically, everything is you use metric. So I mean, that's why you might notice if you look at any incubators or if you hear anything, even temperature we talk about in Celsius. We don't talk about Fahrenheit. You know, bacteria like to grow at 37 degrees Celsius. Ends up being about 98.6 degrees Fahrenheit. Is they kind of like to grow in us. Yeast like it a little colder. They like about 30 degrees Celsius. The fish, they like to hatch at about 18. Zebrafish and Drosophila like about 18 degrees Celsius. So different animals have different happy temperatures. But units of length. So everything starts with that meter. And then you go 1,000th of a meter, gets you down to your millimeter. There is a decimeter in there. It's never used. It's a placekeeper. So, I mean, we usually talk about centimeters, millimeters, or then you jump down again to micrometers. So 1/1000th of a meter is a millimeter, which is about the thickness of a dime. 1/1000th of a millimeter is a micrometer, which is what we've been using to start measuring cells. And then you can even go down further than that. 1/1000th is of a micrometer is a nanometer. And this is what you need to use for organelles or viruses. Okay? So that's 1/billionth of a meter, just to kind of give you some perspective of sizes. Okay? So you'll see a lot is this mu, this kind of backwards U thing for the micro, and then N for nano.
Which is viruses.
Also to help put things in perspective size-wise. I always kind of like seeing where things lay out. Makes things a little easier. So as you're going down, yeah, you have a pig, going for meter, chicken egg, flea, large protozoa. Then we start getting the human blood cells, chloroplasts, mitochondria, start getting some bacteria and archaea. So, you know, our organelles are bigger than some of the prokaryotes. So, start getting your viruses, your ribosomes. Ribosomes are small. They are an organelle, they're small. Proteins. Diameter of DNA is, it's small. I think it's 10 angstroms, so that's about, so you get your amino acids, you get all the way down to your atoms. So just kind of size progression. So, really important. NM, nanometer. The μm is micrometer. And you know millimeter by now. Okay. Microscopes. I know this was a good get to this, but we're going to talk about microscopes today. So, basic principles. We were talking about total magnification, and so this is obtained by multiplying the objective lens
By the ocular lens.
by the ocular lens. So, which one's which?
Ocular is by the
objective.
eye, and that's 10.
Ocular is by your eye. Objective's the one we can rotate. Okay, so we look at total magnification, resolution. So resolution is the ability to see two objects as they separate. I had one person say that it's, let's just go with that. It's as you separate. So it's being able to actually see these are separate objects versus they're close together. So, if you watch something coming from the distance, you might see it as one object coming towards you, and then as it gets closer, you realize, oh no, that's really two cars. And I didn't quite notice that other one because of the resolution. The light microscope, we're down to 0.2 micrometers. So it's like, okay, they're pretty good. They're not great, but we're using light. Contrast. So this is something we were having a problem, especially with the yeast yesterday. So being able to distinguish the specimen from the surrounding background. Okay, so this is where you try and change that contrast. Sometimes dimming the light makes it so it can help. This is why we use stains. We will be using stains tomorrow. So, general principles. You want to look through wavelength, color, visible color. So you have this broad range of colors. And when we talk about fluorescence, what they actually do is you use UV. You notice it's down here. So you shine UV on your sample, and that excites the fluorescent molecules, so then will give off one of these colors of light. So that's how you can get a readable signal, visible signal to our eyes by looking at this. So I kind of like looking at it just because it's reminding me where everything is, where are the waves, what are we looking at, and how small of a range we can actually see. We're talking wavelengths, just in case you're not, one wavelength is from crest to crest or from trough to trough. We use these for light microscopes since that's what's visible. And so you can see that as you use smaller or better, your resolving power gets better. So we don't necessarily have the greatest resolving power because we're using light. That's why for some things we need to use those electrons because we get much greater resolving power. So what are we talking about when we're looking through a specimen? We're talking about light coming in, hitting the glass, and refracting. And so what this does is, with our special lenses, it refracts, makes a focal point, and flips the image around. So you get that inverted, reversed, and enlarged image all in one shot. So this is where you get that magnification. So those different lenses give you different magnifications. So that same basic picture before, but now showing what kind of microscopes you would need to see these. So you can all see a chicken egg or flea. You don't need any help. or hopefully you don't need any help. Light microscope. You start using these, we were seeing protozoans. And if you get really good, you can start, we were able to see some of those bacteria and stuff. And also we'll have that out today so that you can do that again. And then, we won't use this here, but I'll talk about it a little more, is electron microscopy. And so these are kind of cool because you can start seeing the actual protein. So this is one of the newer fields, is where they're actually visualizing the proteins, or this is how you visualize those viruses. So actually being able to see what's going on. Being able to see how does HIV actually attach to the cell. Question? Contrast. So contrasts are difference in intensity between two objects, so the object and the background. It's very important in determining resolution. The higher the contrast, the easier it is to tell apart if you have multiple objects. And this is why we use staining and light that is in phase. So by changing that light, you help to change that contrast. Okay.
What do you mean in phase? What is in phase?
The book has a great picture of it. It's complicated to look at, but it's just because like hitting it at the right spot, so that way you're going to get the most dispersal pattern from it. Versus, because if you crank it all the way up, you're going to have the wavelength is going to be wrong wavelength, so that way you're going to just blank everything out. I know some of you I think had that light way up and you remember hurting your eyes looking through the microscope. So that would be out of phase because you blast everything out. But if you can dial it down, you can get it in phase so you can start seeing that contrast. Does that make more sense?
Yes.
Okay. Microscopes. So you can start getting really good ideas about them. So we have our ocular lenses. So it's for, so you can actually see what's going on. We have what's called the body of the microscope. They move nicely apart, so you can actually make it so you can see through both eyes. It's called the arm, so you always want to make sure you hold it by the arm and the base. We have our coarse focus, our fine focus, our illuminator. And then here's where we have our diaphragm, so it controls the amount of light that can enter the condenser, which is up here. You have your stage, your specimen holder, and this, I guess, on the other side, you have how you can move the stage. You have your objective lenses, okay, basic parts of a microscope. So it's one of those weird things because we're looking at two eyes, they go in, they cross, you're looking at this, it crosses, and it's all because of this light. So you can see the different path that the light's actually having to travel. Okay. Let's make the microscopes make a little more sense. Oil immersion. Why do we do oil immersion? Okay, so when you're when you're doing microscopy, light source, specimen, you're going through the slide, you're going through your specimen, you're going through your glass cover slip, you're going to get some refraction. And for most of it, that doesn't matter. You're being able to see enough that who cares that you're losing some of this. But when you're using the higher resolution, we're so close, if we can get rid of that lost or refracted light, we can see the images better. So that's why you use that oil immersion, which then makes it so that you don't end up refracting it, that you get more light actually coming into that objective lens. Okay? Yeah, light is refracted at the glass air interface, but not the glass oil interface. Types of microscopes. Light and bright field. Okay, so what we're using, the advantages, incredibly easy to use. You guys used them yesterday. For the most part, weren't really any issues. Other than somebody taking the head off of theirs.
Oh yeah.
They're readily available, they're relatively inexpensive, but you can also, as some people found, you can look at air bubbles and think they're looking at cells. So you get that lack of contrast between the cells, and the water. But then again, it's also very easy to stain to make things a little brighter so you can see this. So if we're looking at bright field, you know, you get a series of lenses for magnification. Light passes through your specimen into your objective lens. Your oil immersion can increase your resolution. So this is what you would see. Maybe looking at Saccharomyces cerevisiae, in case you didn't see this yesterday. It is a budding yeast. This is actually, yeah, this is budding here, probably budded here previously. So this, I mean, light microscope is the most common microscope. Phase contrast. So, contains special condensers to help you increase the contrast when viewing an object. This is really good for viewing things without a stain. There's also dark field, which is where you get your objects look bright against a dark background. And so this also, you have to have a special condenser that blocks most of the light. And so this is when you're trying to look for like motility of organisms. So if you're trying to actually watch them. Because sometimes, I mean, those organisms are pretty hard to see, especially when they're swimming. And so if you can really up that contrast, it's a lot easier to track them. So here's an example of phase microscopy. It's great for examining living organisms, something that, you know, would be damaged or altered by putting them on a slide and staining them. Dark phase microscopy. So, yesterday we looked at volvox, or hopefully volvox. You saw them as these nice little rounds, and those were very green structures. But now if we were, so that's looking at them through a light microscope, but if we were looking at them in dark phase, you would actually see them, you know, they'd be darker. So, this increases the contrast, and it makes it so that you're able to see more details. Pale objects are best observed with the dark field microscopy. I mean, those little spots, the pale greens and everything, that really pops with a black background, whereas you can see how those wouldn't really show up, or didn't really show up yesterday, when you have that white background. Okay? So, dark phase, the way this works is you actually have dark field stop. So you have your specimen, you reflect most of the light, so you get this only, you know, the refracted light from the specimen. And so it's just kind of a little weird twist that I want to at least give you a picture of, just because it's kind of an odd concept. Okay. There is also Normarski. So this is differential. And so this is kind of nice because you can really start to kind of see the 3D effects of a sample. Okay? So it really starts to make things pop a little better. Because you're separating the light and then recombining out of phase to produce that better contrast. So, the 4 basic types we just talked about. Bright fields. So here we're looking at a nucleus versus bacterium. You look at it in dark fields. Phase contrast, you see how you start seeing more, and then even more when you see that Normarski. So it depends on what you're looking at, what kind of microscopy you should be using. Okay. That makes sense? Fluorescent. We talked about fluorescent earlier. So fluorescent's a lot of fun. We do it a lot in science. You use this to visualize. So the thing is, you can treat cells with a fluorescent dye. You can tag proteins with a fluorescent dye, so you can actually visualize individual proteins in a cell. You use UV as a light source to make that, you know, to excite whatever's giving you the fluorescence. Another type is electron. We'll talk about in a minute. And so, with an electron microscopy, you actually use electrons instead of light. So since you're shooting electrons at a specimen, actually get, since you have much smaller wavelengths, you can get a lot higher resolution. I mean, we're down to what? 1 nanometer magnification, and the magnification is 100,000x. So you can get a lot better resolution. I mean, we're talking angstroms, which is to the -10 nanometers, -9. So you can get down. Also, some cells are naturally fluorescent, which makes them even more fun because you can just kind of pop them in and look at them. So this is what you would look at, and so it's great for if you're trying to differentiate between the stuff. I mean, if you're looking at pond scum and you want to see your organism, all of a sudden you turn on your fluorescence, shine that UV through there, it's going to, then the proteins are going to transfer it into a visible light, and you see these nice, pretty fluorescent organisms coming up. And another thing is you can actually dye things fluorescently. Something we do all the time in science. You have an organism or you have a protein you want to tag. You have an antibody, which is specific to whatever you're trying to probe. And then you have fluorescent dyes, which are specific for your antibody. So a lot of times you actually use 2 antibodies. You'll use a primary antibody, which is specific to your organism you're trying to stain. And then say you made it in a rat. And so then you'll have a fluorescent antibody that was made in goat, which is specific to rat. And so it's going to just find all of those rat antibodies and bind. And it's how you can amplify your signal, because instead of having 1 fluorescent dye per antibody, you have multiple fluorescent secondary antibodies binding to that 1 primary. But, so you have all those cell surface, those antigens, that's what that antibody is going to recognize. They bind, and you end up with pictures of fluorescent microorganisms. So in this one, the testes. Okay. Questions? Microscopy. Okay, so electron microscopy. Electron microscopes are huge, and they weigh a ton, so usually they're in the basement. UC Davis, I think, has a couple of them, and they're all in the basement, surrounded by lots and lots of concrete. So, okay. Upside down, light microscope, you have your lamp, the condenser, your specimen, objective lens, eyepiece, finally seen. You see that this is reverse of what we would use. But this is what happens with you have your electron gun, condenser, specimen, objective lens, projector lens, and a final image on a fluorescent screen. So it's reverse of how we would do it, but then again, you can't see these with the human eye anyways. You have to take some kind of picture that you can then look at on a computer. So we're looking at these. This is the size of those microscopes. Okay. But you can also get really small. Okay. So, other pretty pictures are there? I'll give you another one of these. Okay. So just kind of more an up close idea. Just so you can see this scanning electron microscope. Just the idea being that it's so much bigger because you're having to use electrons, and then you can get the photo multiplier so you can actually see it in the monitor. But this is how we can see viruses and how we can see organelles and get down to that really small level. Here are the pretty pictures of electron microscopes. So, different things you can see with an electron microscope. I mean, come on, we're at 100 nanometers. So a light microscope cannot resolve structures closer than 200 nanometers. So obviously this is much better. This is for magnifying if you're doing the 10 to 100,000 x. And so sometimes, this is, we can hardly see the bacteria with our light microscope. So you can imagine if we can go even further, how much easier you can see the bacteria, you can see the viruses. There are two basic types. There's a transmission electron microscope and a scanning electron microscope. So, I mean, these aren't very, what do they call, sexy pictures. But then when you start looking at these, those are pretty good. It's kind of cool being able to look up close at what some of these structures are that we would never see this. Okay, these are all scanning electron microscopy pictures. So, it's Streptococcus, paramecium. So, I mean, you're looking at these underneath the slide. You're not going to see anything like this with a light microscope. They're cool, they're useful. You get very pretty pictures. So, scanning electron microscopes, what they do, is you basically just the surface of the specimen is first coated in a metal, like platinum or gold, and then that focuses the beam of electrons back and forth over the surface that's coated. So you're scanning it rather than trying to penetrate it. So that's why you can get this 3D image. It's because you're just kind of trying to see what is on the surface. Yeah.
Is aspergillus a fungus?
Yeah.
Okay.
Okay. Okay. So we've talked about all sorts of different kinds of microscopes. So going back to what we're going to actually use in class, light microscope, and to make it so that we can see things better, we're going to use stains. So stain, the use of a dye to aid viewing. There's simple stains, where you use one dye to add color to cells. So components like crystal violet, so it involves nothing more than basically soaking cells in a dye and putting them on a slide. Okay. We'll also talk about differential cell stains, so using two dyes to distinguish cells on the basis of staining properties. And then smears. Basically what we did yesterday, with your cheek smears. You microbes spread on a glass slide used for staining, you air dry them. We didn't heat fix them, but it's something you can easily do. So, spread the culture in a thin film. We'll let them air dry. This is what we'll do for a lot of for other things we'll do them later, especially if you're going to want to stain them. If you're going to want to stain something, you want it to stay. With everything we were looking at yesterday, other than doing a quick stain and dropping dye in there, but spread it, let it air dry, pass it through flames. We'll get to learn how to play with flames today. Okay. So, this is just kind of that different in contrast of you add a simple stain, all of a sudden it's a lot easier to see than before the stain. So this is with crystal violet. So all you have to do is you soak, the cells take up the stain, rinse them, so that way you don't have purple everywhere. And you get your nice pretty pictures. So, differential stains. Gram stain. Go into gram stain. This is what you're going to be doing tomorrow. So you stain with crystal violet. It's the primary stain. It binds to the cells. You then add iodine, mordant, so the chemical that helps the stain to adhere to the cells. So, flood the smear with the primary stain, then you rinse it away, flood the smear with the mordant, rinse that away. Okay, decolorize with alcohol. So ethanol and acetone, use this as a rinse. Gram-positive cells, purple, what kind of cell wall structure?
Thick.
Okay. Gram-negative cells, they're colorless at this point. And then you counter stain. Okay? So you add that counter stain, flood it, the smear of the counter stain. Now, your gram-positive cells, they're all purple. Gram-negative cells, they're now light pink. Okay? So just to kind of show you what that looks like. Okay? Flood with the crystal violet. Wait about a minute, rinse it with water. You get everything's purple. The different degrees. Flood the iodine. What's the point of the iodine?
See the contrast.
Help it stick to the cells. Okay, it's mordant. Then flood them with alcohol and acetone. So, rinse with the water. You start seeing that some are decolorized. Then you add that counterstain, Ceradon. So, let it sit for a little bit. Rinse it. Blot it. You now can see the gram-negative and gram-positive cells. See the nice pretty pinks and purples? They're better on the computer. I will put this on D2L.